So welcome to the presentation Hardy Weinberg principle and its importance in the evolution. The English mathematician G.H. Hardy and the German geneticist Weinberg independently published a mathematical description of equilibrium gene pools in 1908. So it was Hardy and Weinberg who independently came up with the mathematical analysis of genetic equilibrium, right? And it was for the analyzing genetic composition of populations, and this is popularly known as Hardy Weinberg law or Hardy Weinberg principle, and this forms the foundation of population genetics and modern evolutionary theory. The Hardy Weinberg law states that in a given population, with random mating and no selection between genotypes, the relative frequency of genes or alleles tend to remain constant from generation to generation. In simple words, that the genetic composition of a population, it will remain constant from generation to generation. What are the conditions? The conditions are that in the population the mating should be random and there should not be any natural selection process or any selection pressure on the population. Okay, there should not be any differential selection on the populations, right? So on the genotypes, all the genotypes should have same survival rate. Okay, so if those conditions are prevailing in a larger population, obviously the frequency of different genotypes will remain almost constant from generation to generation, and that condition is known as genetic equilibrium, or the stability of genotypes in populations. It is referred as genetic equilibrium, and genetic equilibrium is maintained when the population is large enough. Okay, when it is small, what happens? Very slight change is going to affect the population. Okay, but if it is large, what happens is it can be compensated. It is not going to reflect much. Okay, so the population should be large. Okay, and there should not be any migration or any subsequent gene flow into or out of the population. Okay, there should not be immigration or emigration that can result in losing of the genotypes. Okay, so there should not be any such kind of gene movement, gene flow. Mutations should not occur, so there should not be any net mutations. Then gametes carrying the two alleles are produced in equal numbers. Okay, that if there are only two alleles for a particular character, the gametes produced should have equal number of gametes should be produced in both the with both the alleles. Okay, then mating should be random. Random mating is a must, and all genotypes, dominant homozygous, then homozygous recessive, heterozygous, all genotypes should have equal survival rate. Okay, and this environment does not change in order to favor any particular genotype. That means there should not be any natural selection pressure on any of the genotypes favoring any of the genotypes. Okay, so all the genotypes should have equal survival rates, all the genotypes should have equal representation while forming the gametes. There should not be any migration, there should not be any mutations, and there should be random mating. Okay, so these are the conditions upon which the genetic equilibrium can be maintained as per Hardy and Weinberg. Okay, under these conditions, the gene frequencies will not change from generation to generation, and there are no changes of variations being produced in the population. So then the rate of evolution will be 0. Okay, so in large populations, evolutionary change can take place only by upsetting these conditions, upsetting the Hardy Weinberg equilibrium. And how can Hardy Weinberg equilibrium can be upset? If these conditions, any of these conditions are upset. Okay, so upsetting the Hardy Weinberg equilibrium can result in evolutionary changes. The factors that can bring about upsets in the gene frequencies are mainly environmental change, mutation, natural selection, non-random mating, genetic drift, migration, and gene flow. Okay, each one we will look into in detail. Okay. When we speak about the conditions for genetic equilibrium, you can see that large populations, small populations have chance can have chance fluctuations in allele frequency. For example, a fire or a storm can bring about change in the genetic frequency, and this can actually disturb the gene frequency in a particular population, smaller population. So, this does not reflect on larger populations. Then migration, immigrants can change the frequency of an allele by bringing in new alleles to a population, or it could even shift the gene frequency to a particular side, okay, in a particular direction, right. Then no net mutations. If alleles change from one to another, this will change the frequency of those alleles, okay. I hope you know what are alleles, right. For example, when you have been studying genetics, you may have written, like, for example, for homozygous tall, you may have written capital T, isn't it? And each of these, it is considered to be allele, right. Allele for tall represented in 2 sets, right. Then capital T, small t, there are 2 alleles, one is for capital T, another for small t, 2 alleles, okay. So, I hope it is clear, right. So, random mating, if certain traits are more desirable, then individuals with those traits will be selected, and this will not allow for random mixing of alleles, okay, and this can upset Hardy Weinberg equilibrium, right. And no natural selection. If some individuals survive and reproduce at a higher rate than others, then their offspring will carry those genes, and the frequency, the genetic frequency will change in that direction the next generation, okay. Now, we will see in detail how the Hardy Weinberg equilibrium can get upset. Okay. First is environmental change. So, environmental change alters the selection pressure, natural selection pressure, okay, favoring a particular gene or genes whose frequency increases accordingly. So, this is the genetic basis of progressive selection. So, whenever there is a change in the environment, it can also bring about change in the genotype. Why? Because the individual have to get adapted to the changing environment, and for that sake they make slight changes in the genotype, okay. And this genotypic change, it can later, for example, there are, just imagine there are 100 individuals in a population, and the environment where they live in, okay, it starts to change a bit, okay, and some of the individuals, say maybe like 20 individuals of the among the 100, 20% of the individuals, they start to develop certain adaptations, and they start to show certain genotypic changes for getting adapted to the changing environment, okay. So, 20% have shown slight genotypic change, okay. Earlier all the 100 had the same genotype, but when there was a environment change, 20% of them started to show adaptations, genotypic change that can suit them to adapt to the changing environment, right. Now, what happens is the nature selects those 20 because they have favorable genotype, isn't it, favorable genotype that can help them to adapt to the changing environment. That is a favorable genotype, okay. So, the nature selects these 20 and starts to eliminate the rest 80, fine. Now, what happened is, earlier there was genotype, just imagine a single genotype, and then when the environment changed, the population 20 got one set of genotype, while the rest 80 had the older genotype, okay. It was different, we had 2 genotypes in the same population, right. Now, when the natural selection started to act upon it, what happened? The 20 with a newer genotype got selected because it had favorable adaptations, favorable genotype suiting to the changing environment. What happened to the older genotype? It got eliminated. Now, what is the condition of the population? The population had a newer genotype. You getting? Okay. So, it is an evolution. So, such environmental change can alter the selection pressure, it favoring a particular gene or genes whose frequency increases accordingly, and this is the genetic basis of progressive selection. I hope it is clear. Okay. Now, second factor that can upset the Hardy Weinberg equilibrium is the mutation. Mutation, it is a change in the chemical structure of gene or chromosome, as we have already seen. And as an evolutionary force, mutation is the ultimate source of new alleles and new gene combinations. So, whenever there occurs a mutation at one or more locus or loci, what happens? There is a gene change, there is a change in the gene or gene combinations. So, there is a obviously the appearance of new alleles and new gene combinations through mutations will produce a change in the gene frequency, right. So, thus the genetic equilibrium is upset, having the like paving the way for the evolutionary change. Okay. The third one is a natural selection. Natural selection favors favorable mutations, or what we call as the useful mutations, which have the highest adaptive efficiency between the population or within the population. Okay. Population, the individuals which can survive well in the environment will get favored. Okay. All other unfavorable genes get eliminated. So, these favorable characters or the genes are then more rapidly spread through the population than others, and that is natural selection removes certain genes from the gene pool while favoring certain others. So, this differential reproduction of genes or spread of genes thus alters the genetic equilibrium, leading to the evolution. Okay. Now, the fourth factor that is responsible for upsetting or factor that influence the Hardy Weinberg equilibrium is the non-random mating. Okay. Gene frequencies in a population will remain constant only when mating is a random process. Okay. Random mating is a process in which any number any member of one sex is equally likely to mate with any member of the opposite sex. Okay. So, in most populations what happens is mating is a selective process and not a random one. Mates are selected on the basis of certain mutually agreeable or desirable traits, which are often adaptive traits for the particular species. So, individuals with undesirable traits are not likely to mate, and so their genes get eliminated. On the other hand, genes representing adaptive traits or what we call as the favorable traits will spread more rapidly than others through the population. So, as a result, the gene frequency will get shifted or altered, upsetting the Hardy Weinberg equilibrium. Clear? So, non-random mating is the normal natural process, but random mating, non-random mating, but the mating only if the mating is random, the Hardy Weinberg equilibrium can be maintained. Fine. The next one is the genetic drift in small populations. Okay, or it is known as a seawall ride. Okay, we will be obviously looking in detail later, but the action of genetic drift was studied by seawall ride in 1931, and hence it is referred as a seawall ride effect, and it refers to the accidental fluctuation in the proportion of a particular allele in a small population, resulting in random establishment of genetic types without being influenced by natural selection. So, we will be looking in detail in the coming presentation. So, here it is genetic drift simply means like accidental shift or fluctuation in the proportion of a particular allele, right. So, there is a shift from one genetic type to the another one, and it usually happens in small population. Okay. Then the last one that influences Hardy Weinberg equilibrium, it is a migration and the gene flow. Gene flow due to migration may also upset the genetic equilibrium, and individuals may migrate from one population to another, and breeding may take place between the individuals over there in the new population. As a result, the genes of one population get transferred to another and vice versa. Such gene flow may result in addition of a gene or loss of a gene or genes, okay, and in the respective gene pools. Hence, the gene frequency gets altered and Hardy Weinberg equilibrium gets upset. Sometimes a neighboring population may join, and if interbreeding take place, they merge into a single population, but with a new gene pool. Okay, so that can also upset the Hardy Weinberg equilibrium. So, we saw what is Hardy Weinberg equilibrium, what are the the factors that can upset Hardy Weinberg equilibrium, and what are the conditions that has to be maintained for the genetic equilibrium to be there. Okay. Now, we can see the application of Hardy Weinberg equilibrium. Okay. Here okay. So, here you can see given this is a condition, this is complete over here. You can see there is a 3 genotypes, one is capital YY, second is capital Y small y, and third is small y small y. Um, it is said capital Y genotype, it is present 49% of the individuals, 49, it is given 0.49, 49% of the individuals in a population is having or are having capital YY genotype. Okay, 42% of the individuals are having heterozygous condition and 9% they are having recessive homozygous condition. Okay. Regarding the individuals, when we speak about the individuals, right, of in a population of 500, 49% means it is 245. Okay, so 245 out of 500 do have this kind of a genotype, while 210 individuals out of 500 do have the heterozygous condition and 45 with the recessive genotype condition. Okay, so we were speaking about the phenotype and genotype. Now we can see the allele, number of alleles. So here in this kind of a homozygous dominant, there are two dominant alleles, right? In heterozygous condition, 50% of the genotype is actually dominant and 50% is recessive, right? So one in this condition, one is dominant allele and the other is recessive allele. Okay, 50% dominant, 50% recessive. And here it is 100% recessive. Both alleles are recessive in nature. Okay. So when we count the number of alleles in the gene pool of 500 individuals, right, we can see that there are 245 individuals with capital Y, right. So how many Y alleles are there? It will be 245 plus 245. Each individual have 2 alleles for dominant character, isn't it? So obviously 245 individuals will be having 245 into 2, it is 490, clear, right? And 210 individuals are having heterozygous allelic composition, right? So what happens is in the 210 individuals, 210 alleles will be dominant and 210 alleles will be recessive. Okay, so we have 245 plus 245, 490. In the population of 500 individuals, how many dominant alleles are there? 490 from the homozygous dominant and 210 from the heterozygous condition. Okay, a total of 700 alleles are dominant. Clear? Coming over here, we can see how many alleles are there. Okay, out of 500, 210 recessive allele from the heterozygotes, 210. Okay, 45 with homozygous recessive character. So they have 2 alleles per person, recessive allele per person. So 45 into 2, that makes up 90. So 210 from the heterozygotes and 90 from the recessive homozygous. So it makes up 300. Okay, so 700 and 300. 700 alleles dominant, 300 recessive alleles. Okay, so when we study allelic frequency, when we have to find the allelic frequency, so out of 1000, 700 plus 300, out of 1000, 700 is dominant. Okay, so dominant allele frequency is 700 by 1000, which makes up 0.7, right? And recessive allele frequency is 300 by 1000, that is 0.3. And you can find that P plus Q is equal to 1. Okay, and this is the same for whatever be the proportion of genotype and phenotype. Okay, it will be P is the dominant allele frequency and Q referring to the recessive allele frequency, and when we add it up, it will be equal to 1. Okay, clear? Now coming to the Hardy Weinberg analysis. Okay, Hardy Weinberg, as per Hardy Weinberg equilibrium, since we have already found P plus Q is equal to 1, obviously P plus Q the whole square should be equal to 1. P plus Q the whole square is P square plus 2PQ plus Q square. Clear? And this one, P square represents, P represents the allele frequency for the dominant gene, isn't it? And P square represents the genotype frequency for the dominant one. Okay. Q represents the recessive allele frequency, right? While Q square represents the recessive homozygous recessive genotype frequency. clear? Okay, that means p square shows the number of individuals in a population having the homozygous dominant genotype. Okay, q represents the recessive allele frequency, that is in a population how many recessive alleles are there, that is what the q represents. Okay, q square will represent the number of individuals having homozygous recessive genotype. 0.09, got it? 0.09. Fine. Now what remains is 2pq. Okay, 2pq represents the number of individuals with heterozygous condition, 0.42, 0.42. Clear? So if we can we can apply the Hardy Weinberg analysis if we know any of this. We needn't even know the whole thing. If we know if we know any of the what you call the genetic genotype frequency of any of these, dominant, completely homozygous dominant, homozygous recessive or heterozygous condition, we can very well find out the number of individuals with a particular genotype, number of dominant recessive, heterozygous, dominant homozygous dominant, homozygous recessive and heterozygous condition. We can find out each one. Okay, we will see with an example. Okay, here there is an example. B is a represents B is a variant form for brown eye color, and small b it is for blue eye color. These are the two genetic variant forms for eye color. Okay, if we know in a particular population we have observed that 9% of the population have blue eyes. Okay, and we need to find the frequency. Okay, the allele frequency, P is the allele frequency, isn't it? The allele frequency of brown eyes and allele frequency of the blue eyes. Okay, along with that we can also find the number of individuals in the population with homozygous brown eyes, homozygous blue eyes and heterozygous condition. We can also find all these. Okay, we will see. So already we have seen that Hardy Weinberg equilibrium, as per that p square plus 2pq plus q square is equal to 1, where p is the dominant allele frequency and q is the recessive allele frequency. Okay, now we have already mentioned 9% have blue eyes, that is recessive genotype. Okay, recessive genotype will be small b, right? So small b is actually q square. P is the dominant allele frequency, while p square is the number of individuals in a population with homozygous dominant genotype. Okay, q is the recessive allele frequency and q square is the number of individuals with homozygous recessive genotype. Fine. So here it is mentioned 9% have the recessive genotype. Okay, only when it is homozygous it will get expressed, isn't it? So 9% with the homozygous blue eyes means blue eyes is a phenotypic character, and if it is blue eyes are expressed, that means it will be in homozygous recessive condition. Okay, so 9% of the population have a phenotype of blue eye color, and obviously the genotype will be homozygous recessive with small b. Okay, and as already mentioned, q square is the number of individuals with the homozygous recessive character, isn't it? So here it is 9%, so q square is equal to 9%, which means it is 0.09. Okay, now we can find q over there, right? So q will be obviously q square equal to 0.09 means q is equal to square root of 0.09, which gives us 0.3, and 0.3 means 30%. So we have already seen that p plus q is equal to 1, isn't it? P plus q is equal to 1. That is why we have got p plus q the whole square is equal to 1. Fine. So here we apply p plus q is equal to 1. So here we know q is equal to 0.3. So obviously p will be equal to 0.7, which means p gives will be 70%. Clear till here? Okay. So we find the homozygous genotype frequency. It will be 0.49. Okay, p is 0.7, so p square is, you can say, (1 minus 0.3 is equal to 0.7, which means 70%. Clear? Now, P square will be 0.7%, that means 0.49, and 2PQ will be 2 into 0.7 into 0.3, that makes up 0.42. Okay, 2PQ means it is the number of individuals with heterozygous condition, that is 42%. Okay, this is, sorry, yeah, 42% of the individuals are heterozygous, 49% are dominant homozygous, dominant this one, and Q square is equal to 0.09. That means 9% is having, already given 9% with recessive character. I hope it is clear. Okay, this is what the Hardy Weinberg principle can be used mathematically to find out the genetic composition of a population. Fine. I hope it is clear. Right. Thank you.